My name is Karen Mölling. You find my email address mölling@molgen.mpg.de in the middle of this foil. This is a slam I presented on the occasion of the Bionale meeting, a biotechnology meeting in Berlin, 7th of May 2019. Now in the following you will hear my talk, and in addition in YouTube we will place the German version of this presentation. The topic is collect phages to kill resistant or multi-resistant bacteria. This will be a crowdfunding, and the banking account number is indicated at the top. Collect phages by crowdfunding to kill resistant bacteria. There are no new antibiotics for the next 20 years within sight. I broke my foot, so therefore I can't really talk to you in front of the audience, but through microphone. But what you see to the left is an open fracture. Good luck for me that I did not have an open fracture, but a closed one. Open fractures are under threat that they are infected by bacteria from the dirt, but also under doing therapy in hospitals by hospital infections, maybe even bacteria which are resistant to antibiotics. This case was shown that many approaches had to be used, including phage therapy, to cure this patient in the hospital of the army in Berlin. The second one is the diabetic toe. It has been published that this can be cured by phage therapy. There are 3 million Americans who suffer from diabetic toes through diabetes type 2. They are under risk that they may have to be amputated their toes or even parts of their feet. Next is shown psoas, bed sores or ulcer. They are increasing with the number of people getting older in our society. Health insurance should also be interested in these approaches. So this is a very important goal. Furthermore, the EU had supported a phago burn, that means phages against burns, open burns, within the framework 7. The data for this have recently been published and can be read in the literature. Furthermore, lung is a target, otitis, urinary tract infections is tested in Zurich at the university and also in Tbilisi. There are about 60,000 dead people in the EU expected to die of multi-drug-resistant bacteria. Furthermore, 1 person per week dies in Berlin since 5 years. Phages are rather harmless. There are 5-fold increased food production if you spray potatoes. Food industry is very interested in using phages. You can also buy salmon in these days in a foil, which normally is even sprayed with phages without you knowing about it, because phages do not require to be declared on the package. There is no GMO, that means no gene-modified organisms involved. You can eat these, you can wash it, and they don't normally do any harm. Phages are viruses of bacteria. To the left you see an electron micrograph with the dots indicating phages around one bacterium. They are natural compounds. They can be dormant or active. We need to use the active ones to kill bacteria. So we have to select those which can do this. Below is shown a picture on about 10 to the 33 viruses and phages which exist worldwide. They are the most abundant and most successful species on our planet. There is a proof of concept shown many times by curing bacterial infections. Cases exist since 100 years or even longer. Tom Patterson is one case described in the literature, also on TV and YouTube. You can get all details of his treatment. Then there are other treatments which you can look up as indicated here. My expertise is based on what? I'm a professor and director of virology at the University of Zurich, and I'm a senior researcher at the Max Planck Institute for Molecular Genetics in Berlin, where I've worked for 20 years before. I wrote 2 articles which you may want to read. One is a German one, viruses instead of antibiotics. It is published in Spectrum in 2017. You can also ask me to send you a reprint. Secondly, there is a paper in English in the journal Viruses, and the title is A Wake-Up Call, We Need Phage Therapy Now. 10 people included their vote for this call within the paper. It is available through open access. Next, phages are no risk. This has been proven in 100 years. 100 years ago, in Pasteur Institute in Paris, phages were discovered. And 100 years later, in 1917, this was celebrated in the 100 years centennial in the Institut Pasteur to remember of this discovery. I was a plenary speaker because we isolated phages and described them from a patient who we previously had treated with fecal transfer. We isolated the microbiome, that means all microbes, and characterized it in the virome, that means all viruses, including phages of this particular patient. This is all published and open for you to the public. But this classifies me as a researcher who has specialized in this field. 100 years phages have been treated in various infectious diseases, and to the left you see a case 2,500 years ago where since then Hindus in the Ganges River swim, bathe and drink the water, and about 1896 already it has been shown that the water indeed contains curative activity, which later on turned out to be phages. So this is not only a religious act, but it is based on real scientific value. No phages are available these days anymore because antibiotics led to application of antibiotics because that's easier to handle. So all phages have been discarded away, and we are not in the position to really fulfill the demand. That's what we want to do with this call. No adverse events have happened so far. If the therapy doesn't work, one can try to use more phages per bacterium or have to use a different kind of phage cocktail. Since 1936, there was an institution founded in the eastern part of the world, which is in Tbilisi, in Georgia, and it exists till today. These had no antibiotics available and therefore stuck to the phage therapy. They are available today, but they are more requests than the institution can answer the demands. We want to collect phages and perform 2 steps. The first step is shown to the left. Either feces we used and described and published can be used as source of phages to characterize the patient. They need centrifugation, purification, amplification, and then the sequence can be determined. Also, sewage can be used under hospitals or the public sewage. This has been ongoing at the ETH in Zurich very successfully. They got 500 milliliter of sewage fluid, and from that they will isolate about 100 phages, which are now presently under investigation at the ETH for food phages. To the right, you see the type, how we want to type the phages, because the phages require a specific interaction with the host. Not each phage is recognized by each host. So there is a key log relationship, and that needs to be taken care of. So we will characterize the phages by using various bacteria. This is a routine test in every microbiology institution, as the one I'm associated with in Zurich, the Institute of Medical Microbiology. This is a routine test shown at the bottom by using petri dishes, bacteria, and the phages. The step number 3 needs to be performed by a doctor on request of patients. but also there has recent something like compassionate trials. But recently there has been a case series permitted in Germany, which allows a controlled study and does not make GMP production an absolute necessity for production of the phages. The phages can now be produced under what is called magistral pharmacy production. And the institutions involved in Germany are BfArM and the Bundesministry of Health, not the BMBF. They are really pursuing this approach. You need either surfaces or even IV has been used in the case of Tom Patterson by IV injection. If there's an LPS contamination, this can be controlled by the doctors, and antibiotics are not obsolete. They are preferentially be combined with the phage therapy. We want to perform crowdfinancing, which is donation-based, and we want to collect phages to kill bacteria. There are 6 most important bacteria published by WHO with the acronym ESKAPE. The cost per phage is about 1,000 euros. We want to publish them open access and prepare a phage bank, including the data. This can be sent out on demand. Patenting is not possible at this stage of the isolation. Magistral preparation in pharmacies is planned in Germany, and compassionate trials can be larger, up to 50 or 70 cases in a controlled study, as I mentioned, BfArM and BMG. We have 6 partners, 2 in Berlin and 2 in Zurich, 1 at the ETH and 1 at the medical faculty at the university. I'm involved with both of these. Then there is a university in Hamburg, a spin-off from the Berlin Institute. They are specialized on phages. Then a colleague is working in Hong Kong, a specialist on biotechnology. One group is in New York, a former co-worker of mine, and also Paris contributes. Why am I calling here? My qualifications are I'm head of virology. I was for many years in Zurich, and I'm a specialist on viruses and phages. I'm a faculty member there and published 250 papers, but I'm not only a scientist. I founded 4 companies and I was advisor for a biotech field for Chancellor Helmut Kohl many years back, also for Berlin Innovation Forum and an advisor for Novartis. I got the Bundesverdienstkreuz in 2018 and the Verdienstorden des Landes Berlin in 2019. You will get a certificate and tax declaration sheet, and you get a book present indicated to the right, Supermacht des Lebens, or in English, is Viruses More Friends Than Foes, or one of the CD shown to the left. But really, please remember, you have to help yourself, and you will help mankind. We all may need phages one day if something in clinics happens and we cannot be cured by antibiotics anymore. If you are lucky, we can call a phage after your name, as indicated here by Merlingii. Thank you. But please remember my email address, merling@molgenmpg.de, and also here in front I'm showing the account number, which is given DE 86 1007 4 times 0 0881 5441 00 Phages and Dahlem Center GmbH. Thank you for your attention.